Hello students, this is Sheila Samuel. Today we are going to discuss about megasporogenesis. What is it? It is the formation of megaspores from the megaspore mother cells. We have already seen the structure of the ovule and its parts. Now let us see how these megaspores are formed. A single-celled archisporium differentiates in the internal cells of nucleus at the micropylar end. And this cell, that is the archisporious cell, it may function as megaspore mother cells or it will divide periclinally, and the outer one we call it as the parietal cell and the inner one, that is the sporogenous cell. And this sporogenous cell may directly function as megaspore mother cells. And this megaspore mother cell enlarges in size and undergoes meiosis and form 4 megaspores or a linear tetrad of megaspores. And one megaspore at the chalazal end is functional, and the other 3 megaspores, they degenerate and disappears, and the one that is functional will give rise to the embryo sac, and this type of formation of embryo sac from one megaspore, we call it as monospore egg. Then you'll find that the nucleus of the functional megaspore divides mitotically to form 2 nuclei, and that is each of those, that is 2 nuclei, one will move to the one pole, other to the another pole, and they will form 2 nucleate embryo sac. After that, 2 more sequential mitotic nuclear divisions takes place, and they will result in 4 nucleate conditions and then into the 8 nucleate stage of the embryo sac. These mitotic divisions, they are strictly free nuclear ones. That means the nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate stage, cell walls are laid down and they give rise to the female gametophyte, or we call it as the embryo sac. This type of embryo sac, that is 8 nucleated 7 cell one, we call it as the polygonum, and monosporic because it is formed from one functional megaspore. Now here, out of these 8 nuclei, you'll find that 6 are with cell walls, and later on they form the 6 cells, and the other 2 nuclei that will form the, also known as the polar nuclei, and that forms the central cell. Now here, just have a look at the picture here. You can just see this is the embryo sac, and this is the chalazal end, and this is the micropyle end. Now here you see that how many cells here? 1, 2, 3, 4, 5, 6, 7, 8. 8 cells are there. That is 8 nucleated, can see that cells are there. But it is a 7 cell stage. How? This is 1, 2, 3. 1, 2, 3. And then 4, 5, 6. And this will together form one cell, that is the central cell. So we call it as 7 cell stage and 8 nucleus, we'll say. And this type of embryo sac, we call it as polygonum. Now see here, the cells that are present at the chalazal end, that is called as the antipodal cells. One that is present at the micropyle end, you can see they are the one is the XII, other one that is the synergids, we call it as the synergids, and they have got a filiform like apparatus. And what is its function? This filiform like apparatus is to guide the pollen grain which contains the male gametes for the fertilization. So this type of embryo sac, we call it as polygonum and monosporic. In 80% of the angiosperms, you'll come across this type of the embryo sac, that is the polygonum. So this is all about, and that is about the structure of it. You can just have how this are being formed. That is how the megasporogenesis takes place. You can just see here, it is the primary parietal cell and the primary sporogenous cell. Then you see that one, this primary sporogenous cell will function as the megaspore mother cells, and the megaspore mother cells, you see, that is, they will give rise to linear tetrads of megaspores, and here you find that which is at the chalazal end will function as the functional megaspores, and here you see that one, this functional megaspores, that is the three, they degenerates, and the other one, that is the functional megaspore, it undergoes first nuclear division, then second nuclear division, then you find the third one you can find. So you are coming across 8 cells, then they get organized. You will find that they are getting organized, and the one you can see the egg apparatus containing the egg cell. Then this is the 2 polar nuclei, and this one you see another 3 cells you can see here. Now see the complete structure here at the chalazal end. You see here they are the antipodals. Then this is the central cell which comprises of the polar nuclei, and this is the egg apparatus which contains the egg cell, and you see the synergids. These are present at the micropylar end. Always make it a point that the synergids are always present at the micropylar end, which contains the egg apparatus, and this filiform apparatus you can see in the synergid. Its main function is to guide the pollen grain containing the male gametes for the fertilization. So this is the development of monosporic polygonal type embryo sac, that is the female gametophyte. So that's all about. Thank you.